ones which create beauty in nature. Okay? Flowering flowers of tulips and daffodils, etc. These have. So this is an example of tulips with the streaks, the coloured streaks, which make it. Those are caused by viruses. Okay, there's another one. As you can see in the pictures, these are streaks, coloured streaks in them. That's actually caused by viruses. Something called the tulip breaks virus. Plus, there's a few of them. These are actually caused by viruses. So that's an example of this is a symbiotic relationship. Okay, it's symbiotic because the flower gives the virus a home, and the virus makes the flower colourful, so the bees are attracted to it and they get pollinated. So it's obviously a symbiotic relationship, but viruses create these nice, beautiful, streaky patterns in flowers that we all think are very decorative. So that's one example of a nice virus. Another example is something called a helper virus. Okay, this is an example where one virus is actually deficient and can't replicate, so another one comes along to help it. So you have a deficient virus which can't replicate, so you get added a helper virus, comes along and helps it replicate, so you then get replication. So it's not one that we normally hear about, but so examples of this are actually the hepatitis D virus, hepatitis D virus, which needs hepatitis B virus that's stuck. Okay, it needs hepatitis B virus to replicate and infect cells. Okay, so D actually needs B to help it. Okay, the B supplies a surface antigen which helps the hepatitis D infect and replicate in liver cells. Okay, so that supplies a surface antigen. Antigen is a bit of protein. Okay, so that's one example of helper virus. Other examples are, I should point out, plant viruses, like ones that make tulips and daffodils colourful, they do not infect humans. Okay, insect viruses also, they don't infect humans. Scientists often work with insect viruses, and they work to do research because they don't infect us. They do not infect humans. They do not infect humans. Okay. So other examples are there isn't. In humans, there are actually quite a lot of viruses present. There are quite a lot present, and I'm not going to go into those in this video because a lot of them actually do cause illnesses, and it's a bigger topic and it's more complicated, but I'm going to mention something else. You get things called lines and signs. Okay, it should all be in capitals actually. That's long interspersed sequences and short interspersed sequences, I think they're called. Okay? These are, I mentioned these because they're actually thought to be the remnants of evolution of sort of viruses and their evolution. They have sequence similarity to superfamilies of viruses. That's why I mentioned them. They seem to be dormant viruses in our DNA, which actually allow DNA to function. Okay, so these are basically dormant viruses in our DNA that help it function. That's why I'm mentioning them. Okay, lines and signs. The lines are, I'll start with signs. Signs, basically they're sort of 100 to about 700 base pairs, okay? About 13% of mammal DNA is made of them, okay? They consist of the sections of DNA with sort of various bits in them, okay? They have a 5' untranslated region and a 3' untranslated region. This is part of the DNA structure. These are sections which are untranslated, they're not made into proteins. 5', 3'. In the early videos I mentioned 3', 5' DNA. Yeah, as I said, they were originally but they share similarity to viruses. I think they're dormant. So there's also so you've also got we also know that in plants there's ×1, and in fungi there's thought to be a 5, okay? One's called tad, one's called lie, one's called curry, one's called deceiver, and one's called ink. Don't worry about these names, they're just names of them, but there's lines, there's 1 in plants and 5 in fungi, okay? In humans, so I was dealing with signs, wasn't I, here? Okay, signs. Lines, okay, there's approximately 100,000 in humans. There's just bits of DNA which help it work, which originally are actually dormant viruses. They seem to be related to viruses. They're what you call retrotransposons. Okay, which basically they work with, the way they work is approximately is DNA becomes messenger RNA plus ribosome. So proteins are made, that then becomes, you then get reverse transcription, and then cycles back to DNA and you get integration. So basically retrotransposons means they work in reverse. It goes from DNA, instead of DNA, RNA, proteins, it goes DNA, messenger RNA, and then back to DNA, okay? Lines. Yeah, as I said, there's 10,000, 100,000 copies, about 8% of our DNA is made of these things, okay? There's also, I should mention, there's talked about 19 viruses in humans, okay? But I'm not going to go into much more details, it's a bit complicated. So examples of, I mentioned before the helper viruses. So an example is you get a replicated virus which cannot replicate in its own, okay? So it's kind of made up of, it's got genes like this, GAG, envelope gene, okay? And then, so that represents the DNA, okay? And it's got various genes for the capsid, genes for sort of the structure of the virus, but it's missing a gene to replicate called polymerase. But then you have the helper virus too, okay? Which is, it has GAG, pol, envelope, okay? And that means that that's the DNA. That means that you then get proteins made, okay? Okay? So, yeah, I mentioned lines. In humans, about 100,000. I also should mention that lines are I think it's been an estimate of sort of roughly about yeah, approximately 100,000. So I mentioned signs. So lines are long interspersed sequences, and signs are short interspersed sequences, okay? These are these come from RNA polymerase 2, and these come from that comes from RNA polymerase 3, that comes from RNA pol 2, okay? That's RNA polymerase 2 and RNA polymerase 3. Yeah, so as I said, they're originally part of a big virus superfamily. Lines are also I think I mentioned there's about 100,000. They're also approximately about 6,500 base pairs long. Okay, so yeah, the signs were 100 to 700 base pairs. Okay, lines are much longer, but they are called long interspersed sequences. They're much longer. Okay, so there are lots of them. But basically, signs and lines are sections. These are short these are short stretches of DNA interspaced throughout the genome to help it work, and these are long pieces of DNA that are interspaced throughout our genomes to help it work. Okay, and they're thought to come from viruses originally, but they're dormant. So they're kind of what you might call nice viruses. They help it function. They help our DNA work. Okay. So yeah, hope you found that. Yeah, I think that's quite exciting. There's not all viruses are dangerous, so and they certainly don't all cause diseases. Some are very helpful to us. Some allow other organisms like flowers to actually survive and breed. Some viruses help others to actually work, like helper viruses. Plant viruses and insect viruses do not infect humans and other animals. Not all animal viruses even infect humans. You get viruses from dogs that don't come across to us. Like, I don't think coronavirus it's barely infected dogs during this pandemic. But there are some animals that have viruses that don't come to humans. So, anyway, have a good afternoon. They were defining nice viruses once using technology for medicine and so on. That's not what I was quite thinking about nice viruses. I was thinking ones which actually maybe help other organisms. So